BODIPYs that can be used as fluorescent molecular rotors. So we're going to talk about the concepts and obviously about the applications. So let's start with the definition of a fluorescent molecular rotor. So it's quite complex to pronounce, so I'm going to call this FMRs, if you don't mind. So an FMR is either a natural or synthetic fluorescent molecule. The idea is that is composed of two parts. So you have the stator that you can see in blue here, which is basically the biggest part of the compound. Physically speaking, it is the one with the highest moment of inertia. And you have the rotor, that is the red part here. And those two parts are, well, bound to one to each other with a single bond and can rotate relatively freely as it is possible for any single bond to rotate. However, so you have some examples with double bonds, but in terms of photophysics, it's quite different. So we're going to stay with this example of a single bond and just know the fact that, well, an FMR and a molecular rotor in general is two parts of the same molecule rotating towards each other. So you have a lot of different kind of examples. So here you have the infamous BODIPY. BODIPY is actually this core, and you can see here that you have the two parts. So you have the rotor. So basically, this one should be the stator, and this one should be the rotor. So in certain cases, molecular rotors can be uh, symmetric. It is the case with this gigantic, or actually big, synthetic compound composed of two zinc porphyrin units, and that can freely rotate around this bond between the two alkyne moieties. So in terms of pure photophysics, you all know that in general, the UV excitation, or actually light-induced excitation, can lead to two different kind of events. So you have here you go from this, so from the ground state, so this S0 single state. You go up to S1, so the first excited state, or S2, so you have the same spin state. And you can have different types of events. So you have non-radiative events, so it can be vibrational, relaxation, or free rotation. That typically goes into the zigzag type of mechanism. And you have radiative events, typically fluorescence or phosphorescence. So fluorescence in general is quite fast. So um, basically between 10 to the power of minus 10 to 10 to the power of minus 7 seconds. Phosphorescence in general is slightly more complicated. So you need to go from a singlet state to a triplet state through an intersystem crossing. You change your spin state, then you then go to phosphorescence, which are way longer events and radiative events, typically up to seconds, 10 seconds type of phosphorescence lifetime. In low viscosity media, for fluorescent molecular rotors, the rotation of the stator and the rotors can actually occur quite easily. You don't have a lot of constraint. Therefore, you actually have a majority of vibrational relaxation or free rotation, so non-radiative events. And as it's pretty much an equilibrium between these different events, radiative and non-radiative, you have low fluorescence intensity. So your compound would be fluorescent potentially, but actually with a very low intensity. However, if you go to high viscosity media, well, you will have a physical constraint from your medium to your compound, and that physical constraint will hamper the rotation. So you will have less non-radiative events such as free rotation and higher fluorescence intensity. So this is related to an increase in the fluorescent quantum yield and an increase in the fluorescence intensity. So the easy thing to see here, you have a low viscosity, you therefore have a high free rotation of the two units, low fluorescence. You increase the viscosity, you have high fluorescence because you hamper you definitely hamper the rotation. Um, in terms of equation, fluorescent molecular rotors are kind of dictated or should obey to a bunch of equation, and especially this one, which is the Foster-Hoffman equation that tells that for an FMR, fluorescent molecular rotor, the quantum yield of fluorescence should be directly proportional to the viscosity with, so eta here with a power that is actually a constant. So if you take the log of this equation, decimal log, you see that the decimal log of the fluorescent quantum yield is directly proportional to the decimal log of the viscosity. If you go a little bit further, you know that quantum yield is actually the fraction of the rate of radiative decay over the rate of all type of decays, and is also equal to the product between the rate of radiative decays and the lifetime of fluorescence. Therefore, if you inject this equation to this one, you will see that the lifetime of fluorescence is directly proportional to the viscosity. And in terms of physical, physical data, you can see definitely in terms of fluorescence intensity that once you increase the viscosity, you increase the fluorescence intensity. And this is directly related to this one. This is a lifetime, so fluorescence lifetime imaging plot. And you can see that once you increase the viscosity, you do also increase the fluorescence lifetime. And if you have a compound that you have been evaluating as a fluorescent molecular doors. If this compound actually fit these equations, you can say, well, now I know that it's a fluorescent molecular rotor. A very good example to actually illustrate the physical constraint that is imposed by the viscosity of the medium is the comparison between these two BODIPY units. So you have this one here that is basically just a phenyl unit on the BODIPY. So you can see basically there are not a lot of steric constraints between these hydrogen and these hydrogen. And this one in a low viscous medium can rotate quite easily, and you have therefore a pretty low fluorescent quantum yield, so a low fluorescence intensity. However, if you induce some and you increase the sterical constraint that you have and actually hampers the rotation, you will have a compound that will not rotate so much and that will favor the radiative decays such as fluorescence. So you can see that here the fluorescent quantum yield goes up to 0.93, so gigantic fluorescent quantum yield. And that actually illustrates the fact that in very viscous medium, these these solvent molecule can actually play the same role as these methyl here. They basically block and just hamper the rotation. So the applications of FMRs are quite numerous. So they can be viscosity probes in cells, cell membranes, organelles, and actually synthetic membranes. But they can also be fluorogenic reporters of protein assembly or interaction with nucleic acid. This is pretty interesting because you have to understand that once you have a proper interaction between a compound and a target, such as the catalytic sites or any type of site in a protein or a nucleic acid, you will kind of lock your compound in a certain conformation. And therefore, for fluorescent molecular rotors, you will actually block the rotation of the rotor from to the stator. So you will basically just increase the fluorescence and you will be able to localize and characterize your interaction and localize your protein compound interaction. So in terms of applications, you have these very nice pictures. So you can see here a very typical one with the BODIPYs with the doubly charged chain is their interaction in the bilipidic layer, the plasma membrane. And you can see here that with fluorescence lifetime imaging microscopy, you can definitely locate the compound directly on the membrane. You have these gigantic synthetic compounds, such as this one, that are very interesting. they're the best viscometer around, I think, and they have the possibility to be accumulated near nuclei. And you have also these ones. So you have these two uh, porphyrin, zinc porphyrin units, and these ones are quite interesting because they're absorbing in well, deep red or actually near infrared, which is interesting if you do two photon. If you also if you do single one photon microscopy and you want to go a little bit further in deeper in tissues. So now let's go to BODIPYs, which is kind of the core of the presentation here. So this very nice BODIPY name comes from this terrible UPAC name, so 4,4-difluoro bora diazoindocene. They also called boron dipyrines. BODIPYs are this unit. You have the boro, difluoro, and dipyrines here. They're easily functionizable, pre-functionizable on pretty much any of these sites. But the most important part for fluorescent molecular rotors research is the substitution at the meso position. And you can see that it makes a lot of sense to imagine that this this site would be preferable if you want to create an FMR. What is even more interesting is as the core of the BODIPY is quite small, um, substitution with different type of heavy atoms or functions will definitely and very easily change the photophysical properties in terms of um, emission. And you can see here on these four examples that if you go from iodine to a double bromine to a double iodine substitution on the position number 2, you will definitely completely change the fluorescent emission properties from a very, very deep, very profound green to a relatively deep orange. What is interesting was actually fundamental in body PS FMR is if they fit the Foster-Huffman equation and the log of this equation, well, you can say now I have a fluorescent molecular rotor and use it as such. The applications of BODIPYs, so we talked about viscosity probes. They've been used in numerous live cells, lines, organelles, but also as simple ligands. So ovarian carcinoma, um, um, breast cancer cells, lysosome mitochondria, RNA and DNA ligands. They've been used, and it's a very nice study, as very efficient DNA origami ligands to do DNA paint, for instance. So we're going to have a little look at two case studies to see a little bit how you can actually design, use, evaluate, and use BODIPYs. So this one dates back from 2017 and my time at the Imperial College. And it's a good example of how you actually now how BODIPYs are employed and how in general that you work with a fluorescent molecular rotor. So here the target of the study was to study the sensitivity and the correlation of FMR towards viscosity, temperature, and polarity. Uh, it is important because FMRs are usually evaluated towards with viscosity, but as you may know, if you change the temperature of a certain solvent and a certain viscous solvent mixture, you will change the viscosity. And if you change the solvent mixture, you will also change the viscosity and the polarity. So it was pretty important to characterize FMRs, knowing that all these data are actually interdependent. So the workflow of these studies in general are the design and the synthesis of BODIPYs and FMRs, correlation study, viscosity, temperature, and polarity, and finally lifestyle imaging. So the team designed and synthesized 6 types of BODIPYs, 6 molecular rotors. So you had a characterization. What is important here is that it's actually a good example of how you do BODIPYs rapidly, efficiently, and in a few steps, because it was important for me to show you a little bit the type of tricky steps that are involved in these type of studies, especially because if you see this compound, well, you can do chemistry by design, but without knowing the complexity of the steps, it becomes complicated to carry a study. So to form these units here, you will do simple alkylation, and here comes the funny part of the BODIPY core formation. You condense pyrrole you condense actually the aldehyde on two pyrroles. You form this very nice dipyrene core. You have an oxidation here that will form this double bond, and well, you will form the BODIPY core using boron trifluoroetherate, which will have instantly the effect of making this compound very fluorescent. So it's actually kind of the nicest part of synthesis, because your compound becomes very fluorescent right away. And finally, to install this quite complicated, quite nasty actually, doubly charged ammonium chain, you will do a succession of Hoffman alkylations, and you exchange the iodine for the chlorine, actually the iodide for the chloride, through a ion exchange Dweck-Scully. So in the end, you have a compound that is synthesized with not a good yield, but relatively rapidly. And these pathway can be employed and is usually employed in all the synthesis of these body dyes. So we then have a characterization saying, well, are they molecular rotors or not? And we developed a model to explain the correlation between viscosity, um, viscosity, temperature, and polarity. And actually, it was kind of a cool story to just target the compounds that are relatively that give the same type of properties and the same type of lifetime of fluorescence with a fixed viscosity, regardless the temperature and actually regardless the polarity of your compound. Because in the end, it is important to say that if you have a compound that you consider to be a viscosity probe, that well, it would give you the right viscosity through the right fluorescence lifetime, regardless the temperature of your environment and the polarity that can be very varying um, in nature. So what you can see is like this one, for instance, the body P1 that is related to this one. You see that it's quite frantic, because at a certain viscosity, you will have one lifetime. But if you now switch from another type of polarity, you will have another lifetime. So that's not what we're looking for. We're looking for actually properties such as this one, saying that whatever the temperature, whatever the polarity of your mixture, if the viscosity is given, it will give you a lifetime, and and vice versa. Actually, this one is actually the same, but for low viscous compound, which is something that happens quite often in nature. It will give you a range of lifetime which is quite big. So this one is actually the best candidate. So it's compound number 3. It was the same properties for this one, which is very interesting also. And well, this one was selected as being the most useful one, and most useful because, well, now you can actually do a calibration for FLIM, so fluorescence lifetime microscopy, imaging microscopy. Um, and you end up with a compound that will say, well, okay, I have this lifetime, therefore I have this viscosity at this precise position. And so in the end, you do live cell imaging. So on the left, you will have a simple fluorescence imaging, and on the right, in the picture F, you will have a fluorescence lifetime imaging, which is based on what you call a uh, total photon count, TSCCP, which is independent of the concentration of your dye at a specific position. And what you can see is, even though you have a bit of penetration uh, through the membrane, this one should be located at the membrane. You have a bit of penetration in the membrane. What you can see is that you have a relatively heterogeneous viscosity in this SCOV3 cells, which is cool. A second case study, which I quite like, is relatively recent, from 2019, published in ChemCom, and with someone that we actually know pretty well at Idylle, Maïkolo and Andrei Klimchenko, um, based on design of BODIPY-based apoptotic markers, which bind selectively phosphatidyl serine. The workflow was pretty much the same: design and synthesis of a BODIPY fluorescent molecular rotors, the evaluation as a fluorescent molecular rotor, and in the end, lifetime imaging. So the study is quite smart.
Nicolas,
We
am I the only one who only sees the screen is frozen for you guys, or am I the only one?
I see an image with several colors and lifetime in picoseconds.
Okay, so I
Oh,
think the screen is frozen. We are still on slide 21.
oh, I'm slide 24. Okay. I'm gonna go back to.
For
Okay,
me, the connection is really, really bad. In
Okay.
fact.
okay. I'm gonna just go back to this and represent to represent your entire screen. Okay, this. Okay,
Yeah, much better. This we
much
didn't
better.
see.
Okay, very good. So this study by Klimchenko and Mayelkolo is based on, so design of a fluorescent molecular rotor that can bind phosphatidylserine, that is normally exclusively located in the inner leaflet of bilipidic layer, so the cell membrane. So you have the outer layer. I'm gonna no tech. I'm gonna go back to no. So you have the outer layer of the cell and the inside of the cell, the cytoplasm, that is actually here. And so before apoptosis, you have the PS, sort of phosphatidylserine, exclusively located at this point. However, in the early and late stage of apoptosis, you will have enzymes which are called scramblases that will invert and actually locate some of the phosphatidylserine on the outer layer and expose PS on the outer layer. And this is an idea that if you want to detect apoptosis and early or late apoptosis, you will need a fluorescent reporter of this mechanism, the fact that a phosphatidylserine is actually now exposed to the outer layer of the cell membrane. So they realized that they can have a zinc ligand that can bind this anionic and so negatively charged compound, phosphatidylserine, coupled with a fluorescent reporter that will become very fluorescent at the moment that is in contact with something like the bilipidic layer. And they realized, this can be easily done with a fluorescent molecular rotor coupled to something that will bind phosphatidylserine. So they start with this relatively complex, actually, body piece structure, very close to what we had earlier, with a long peg chain that will interact with this part with an azide moiety with which you can do click chemistry if your zinc ligand is equipped with an terminal alkyne moiety. So you do your carbon click chemistry, you form the triazole, and you add some zinc, you have the zinc complex here, and you have a proper zinc ligand that can bind phosphatidylserine coupled with a fluorescent molecular rotor. Well, actually, something is supposed to be a fluorescent molecular rotor, or actually thought to be that way, and which, upon contact and actually upon interaction with the bilipidic layer, will start to fluoresce like crazy. So in order to prove that it's a fluorescent molecular rotor, you have actually to just do classic evaluation. So you realize first that, well, if you start from a low viscosity, you have a very, very low fluorescence intensity. But if you increase the viscosity, you end up with a very high fluorescence intensity. The same type of experiment can be done with the fluorescence lifetime. And it's pretty clear that when you increase the viscosity, lifetime becomes very long. If you plot these two data, so quantum yield and fluorescence lifetime, so if you do the log, you have a relatively good correlation. And you can say, well, yes, it fits the Foster-Rifman equation. It's a fluorescent molecular rotor. So in the end, prove this. And live cell imaging gave something very, very satisfying, knowing that, well, this compound, so the BPDPA zinc, so BODIPY something zinc, was actually very bright in these cells at early apoptosis, late apoptosis. What they do is they, at the same time, well, use a dye called annexin 5 that can actually report and target early and late apoptotic cells. And when you do the overlay, you realize that you have a perfect overlay between these ones, saying that, well, it's the apoptosis tracker activity is actually confirmed. And once you realize and you go for bright field, you realize that there are other cells that are not apoptotic that are not targeted with this BDPA zinc. So, well, target confirmed for this paper. So pretty cool paper, saying showing that, well, you can use your body piece as a viscosity probe, but you can actually use it as a simple fluorogenic. Use this FMR and the fact that it will be very fluorescent in highly viscous medium, but use it as a fluorogenic marker for something else. So take a message, conclusion. So the BODIPY, very cool molecule, very nice, very, very versatile. You can do a lot of things with this. It's a great platform as a fluorescence molecular rotor core. You have numerous applications. So we talked about viscosity probe, but we also talk about that it can be used as a fluorogenic reporter. And we didn't talk about a lot of things, but however, I think we covered a bit of the field. So one of the current development of the field is actually going to, red, deep red. So to use it in do some deep tissue imaging, which is relatively complicated now because the absorption spectra is obviously for this compound is quite small and not very rich in heavy atoms. That is actually centered around 350 to 450. So if you manage to have a compound that absorbs in the red, you will be able to do some deep tissue imaging. All right, so that's it for me. If you have some questions, I'll be more than happy to answer.